Good morning. So this is the work that together with the Origin Water International. I think Jing is not here. So I think I'll skip this because everybody knows linear economy and the circular economy. So I see this is the basis for our research here. And actually what we were doing is as you heard from Sean and Stefano, we're trying to convert urine into as much as possible fertilizer. So the problem is, of course, you know that when urine is hydrolyzed from urea to ammonia is volatile, and then pH increases, and then also micropollutants are there, and there's a lot of smell problems. So this is what we're trying to be solving the problem through the use of membrane bioreactor to firstly to convert ammonia into nitrate through biological nitrification without having to use any chemicals, and also of course try to sanitize it so they make it safe for reuse as fertilizer. And then we use that. So we're using membrane bioreactor means we use ultrafiltration membrane because that can reject most of the bacteria and virus. And of course, we try to degrade any organics that's present. And of course, then it removes the unpleasant pungent smell from the ammonia. So of course, we are able to recover completely the NPK fertilizer, but there's still challenges. They not able to remove pharmaceuticals and the micropollutant that's present in that. And also this high ammonia concentration, as Sean was mentioning earlier, HRT also impacts. The longer the HRT, of course, it treats well, better. We have a higher conversion rate from ammonia to nitrate. But the problem is you increase your capital, the size of the treatment plant. So that is adding to the cost because of the extended hydraulic retention time. So you are seeing how we can reduce this footprint. So one area we've been doing is to see how activated carbon by powder activated carbon, if you can put it there inside the reactor. We don't do much, but just put it inside the reactor and see how it improves. So as you can see from this, okay, so this graph here, so the orange one, it's is the MBR alone without activated carbon, and the other one, shaded one, is with the activated carbon. As you can see, it's the removal efficiency is much higher, and so the TOC removal is also higher. So the conversion rate is much higher. So that the micropollutant removal is 99%. So if you use just the normal MBR, it's only about 45 to 98%. Some of them are not like less than 40% removed. But as you add the micropollutants, then the PAC, then it significantly improves. So in that way, we don't have to do much in terms of capital costs, just adding a little bit of things helps it. So this is one of the findings. I think this we have already published with Jade Jing as the first author. So here, and then in the second approach, what we tried to do was now, in the first approach here, when we added the PAC, we were able to solve the micropollutants problem, but the other problem still remains, the extended aeration. So hydraulic retention time doesn't really impact by adding the PAC, because PAC concentration we had to control. No, we didn't put a lot. Now, what we did was, okay, we tried to put something here so that we incorporated biofilm barriers there. So we have this plastic bits that commercial one. So we put it there. What the idea is that it can provide the surface area for more bacteria to grow inside, and then in the process, may be able to accelerate the nitrification, and then so in that way we can reduce the HRT. So when you reduce HRT, of course, then we have lesser footprint. So from here you can see that by incorporating this the biofilm carriers, we're able to reduce improve the nitrification rate, like 36% improve the nitrification rate. So that is a significant the reduction in the hydraulic retention time. So hydraulic retention time is around 40%. So from 10 days, say for the week, to able to reduce to around 6 days. So that's one of the achievements that we're working into for reducing this definitely, but this is just the initial, the experiments that we've been doing. Then third, the research progress that we've been doing is the trying to understand how the temperature and the amount of dissolved oxygen impacts the biological nitrification process for the urine treatment. So we did initially earlier studies, but this one we tried to delve in bit more detailed. And then so you can see from here we tried it 10 degrees Celsius, 20 and 30. We didn't do it much because it's usually requires a lot of reactors. So only we did 3, and then we just mentioned in terms of nitrification, the rate of nitrification and the hydraulic retention time. So as you can see from here, actually, for 10, 20, and 30 degrees Celsius, you'll see that here only most of the time it is the 20 degrees Celsius is the quite optimum. And even if you try to increase the DO, say beyond 4 milligram per liter, it doesn't really, it just slow, in fact, it reduces the nitrification rate. So it's no point doing that. So from here, it is, the normal temperature is quite efficient for nitrification rate. And also HRT, you could see up to here. Of course, we're planning to do more than this one, like 8 days. But yeah, you can see that because we don't have the data in between, so it could significantly reduce the HRT. So from here, you can see that increasing or decreasing the pH, the temperature doesn't really impact doesn't really improve the process. And the same thing with the dissolved oxygen. So of course, this is some of the results from the test applications of the urine fertilizer. So it's from here, I can summarize that, it's basically is almost comparable to the commercial hydroponic fertilizers with our urine. So that's the end of my presentation. Thank you very much. Very much, Sherub.